We begin our study of the integumentary system with a detailed examination of its major organ, the skin. The two main components of skin are the epidermis and the dermis. We will spend several lessons examining the anatomy and physiology of the epidermis first, before we discuss the dermis in any detail. The prefix epi in epidermis is frequently used in biological terms, and here means on, above, or outer. Thus the epidermis is on, or above, or is the outer dermis. Make no mistake, however, the epidermis is a completely distinct tissue type from the dermis. The dermis is made of connective tissue, while the epidermis is an epithelial lining. Recall from modules 4.2 and 4.3 the 5 characteristics of epithelia. They display cellularity, polarity, are attached to a basement membrane, are avascular, and are highly proliferative. In regard to their avascularity, the epithelial cells of the epidermis depend on the blood vessels of the dermis to deliver nutrients and gases like oxygen to sustain them. Given that the epidermis is a stratified squamous epithelium, only the deepest layers of the epidermis, which are closest to the blood supply of the dermis, receive sufficient nutrients and oxygen to keep them alive. The most superficial layers of the epidermis do not receive sufficient supplies and are dead. Skin is classified as thick skin or thin skin. Most of the skin on our body is thin skin. The skin on the palms of our hands and soles of our feet is thick skin. There are a number of differences between the two, but for now, most notable is that the epidermis of thin skin has only 4 distinct layers, whereas thick skin has 5 distinct layers. Moreover, the most superficial layer in thick skin, that is the stratum corneum, is substantially more abundant. We will use thick skin to describe the characteristics of each of the 5 layers of the epidermis. Let's look at each of these now. We begin with the stratum or stratum basale. The deepest epidermal layer is called the stratum basale or stratum germinativum. The stratum basale connects to the basement membrane. Before we discuss the stratum basale any further, it will be helpful to clarify the structure of the basement membrane. There is some confusion at times over what exactly the basement membrane is. Part of the confusion is due to several terms that are sometimes used interchangeably, even though technically they are distinct structures. For example, the term basement membrane and basal lamina are often used interchangeably. Technically, there are some important distinctions to be made. More confusion arises because of the adjective basal, which is used as part of a compound name for several distinct structures found here. The adjective basal simply means relating to, or situated at, or forming the base. So we have terms like basal surface and basal lamina, which are distinct structures, but not surprisingly, as their name suggests, relate to or are situated at or form the base of some structure. Let's look at each of these terms to make sure we are clear. 1. You may recall that epithelial cells can display polarity, in that their cells can have distinct poles or ends of their cells. For example, facing the external and or internal environment, epithelial cells can have an apical surface. Along the sides, we have lateral surfaces. And at the base, we have a basal surface. It is common to see the basal and lateral surfaces combined in the term basolateral membrane. Again, this is just a plasma membrane of the epithelial cell. 2. Anchoring epithelial cells to connective tissues as seen in all tissue membranes is a structure called the basement membrane. The basement membrane is simply a thin, fibrous, extracellular matrix that separates the lining of epithelia from their supporting connective tissue. The basement membrane is not actually made of cells. It is made by cells of the epithelia above and the connective tissue beneath. Here, that is the dermis. The basement membrane can be viewed in most tissue membranes using a light microscope. 3. When we view the basement membrane under a much more powerful electron microscope, we can actually distinguish layers produced by the epithelial tissues from the layer made by the connective tissues. Thus, the basement membrane can be divided into several subparts. The layers produced by the epithelial cells are a clear layer called the lamina lucida and a denser layer called the lamina densa. It is unclear if the lamina lucida is an artifact of microscopy or a truly distinct structure from the lamina densa. Regardless, the lamina densa is called the basal lamina. And for simplicity, you can include the lamina lucida here as well as part of the basal lamina. Thus, the basal lamina is one part of the basement membrane, namely the superficial component made by epithelial cells. The final component of the basement membrane is a reticular layer, the lamina reticularis, or fibroreticularis, produced by the connective tissue cells of the dermis. The deepest epithelial cells grab onto the basement membrane via integral proteins that are part of their hemidesmosomes. Hemidesmosomes attach the cells of the stratum basale firmly to the basement membrane. The basement membrane separates the epidermis from the connective tissue of the adjacent dermis. Taking a broader look at the skin, the interface between the dermis and the epidermis is highly convoluted with ridges and valleys. The valleys are called epidermal ridges, which extend down toward the dermis. Between these ridges, dermal mounds or hills called dermal papillae project upward into the epidermis. The function of these ridges and papillae is to increase the area of contact between the two regions and strengthen the bond between them. It also increases the available surface area for diffusion of nutrients between the dermis and epidermis. These projections manifest themselves all the way through the epidermal layers to the surface of the skin. The superficial ridges on the palms and soles, which overlie the dermal papillae, increase the surface area of the skin and increase friction, ensuring a sure grip. The ridge patterns on the tips of fingers are also the basis of fingerprints. These ridge shapes are determined partly by genes and partly by the environment inside the uterus during development. That is, during fetal development, contact with amniotic fluid, another fetus in the case of twins, or the uterine wall will affect the fingerprint pattern of a fetus. For this reason, identical twins do not have identical fingerprints. Then, the pattern of your epidermal ridges is unique and does not change over the course of a lifetime. The major cell type found in the stratum basale are large stem cells called basal cells or germinative cells. Their continuous division replaces cells that are lost or shed at the epithelial surface. There are other types of cells found here as well. You will recall from Module 4.2 that epithelial tissues provide sensation because they are innervated. Specialized epithelial cells that are sensitive to touch, called Meissner's corpuscles, respond to physical stimuli and activate neurons which communicate that information back to the central nervous system. These are actually found in the dermal papillae just proximal to the stratum basale. Furthermore, there are also specialized cells that are squeezed between or deep to the basal cells called melanocytes. Melanocytes synthesize melanin, a red-yellow to brown-black pigment that colors the epidermis. We will discuss the function of melanocytes in a later lesson of Module 5. Beyond such highly specialized cells as these sensory epithelia and pigment-producing epithelia, 90% of the cells of the epidermis are keratinocytes or keratinocytes. These epithelial cells are called keratinocytes because they produce the protein keratin in abundance. There are three distinct multicellular layers between the stratum basale and the superficial layers of the skin. Let's look first at the stratum spinosum. Each time a stem cell divides, one of the resulting daughter cells enters the stratum spinosum, or spiny layer. This stratum, which is typically between 2 to 4 cells thick, consists of keratinocytes held together by desmosomes. They are called the spiny layer because of their spiny appearance, which is due to shrinking of the microfilaments between desmosomes that occurs during staining for viewing with a microscope. They do not actually appear spiny otherwise. Cells of the stratum spinosum are alive, and they may continue to divide and add to the thickness of the epithelium. Apart from keratinocytes, we also find specialist cells called dendritic cells or Langerhans cells in the stratum spinosum. These Langerhans cells, or dendritic cells, take up and process microbial antigens to become fully functional, antigen-presenting cells that aid in the immune response. The dendritic cells are actually found in multiple layers of the epidermis and even the papillary layers of the dermis, but they are most numerous in the stratum spinosum. The stratum granulosum, or grainy layer, is very thin, only 1 to 2 cells thick, and consists of cells displaced from the stratum spinosum. The cells of the stratum granulosum have stopped dividing and have begun to make large amounts of the protein keratin. The cells contain granules packed with proteins that are involved in keratin filament formation, hence the granular appearance. Cells of the epidermis begin to die in this stratum. Keratin is extremely durable and water resistant. In humans, keratin not only coats the surface of the skin, but also forms the basic structures of hair, calluses, and nails. In various other animals, it forms structures such as horns and hooves, feathers, and baleen plates in the mouths of filter-feeding whales. In the thick skin of the palms and soles, a glassy stratum lucidum, or clear layer, covers the stratum granulosum. The stratum lucidum is typically 2 to 3 cells thick. The cells in this layer are flattened, densely packed, and filled with keratin. Finally, at the exposed surface of the skin is the stratum corneum. In thick skin, it normally contains 15 to 30 layers of flattened and dead epithelial cells packed with filaments of keratin. Such cells are said to be keratinized or cornified. The dead cells in each layer of the stratum corneum remain tightly connected by desmosomes. As a result, these cells are generally shed as sheets rather than individually. In total, it takes between 7 to 10 days for a cell to move from the stratum basale to the stratum corneum. During this time, the cell is displaced from its oxygen and nutrient supply, becomes filled with keratin, and finally dies. The dead cells usually remain in the stratum corneum for 2 more weeks before they are shed or washed away. As superficial layers are lost, new layers arrive from the underlying strata. Thus, the deeper layers of the epithelium and underlying tissues remain protected by a barrier of dead, durable, and expendable cells. Normally, the surface of the stratum corneum is relatively dry, so it is unsuitable for the growth of many microorganisms. Join me in Module 5.2 as we examine how pigments in the epidermis and blood flow in the dermis influence skin color.